Evening. I hope you guys are doing well. Welcome to Digestion Week 9 for Biology 1022. My name is Ian Coates, and I'll be your facilitator for this class as through weeks 1 to 8. We're getting down to the nitty-gritty now, so please don't forget that I'm going to go right through, but I encourage you guys to stop and start this whenever you can. Take your breaks. Read the textbook, and you guys will be just fine. Let me know if you guys have any issues. Other than that, have yourself a great class. Talk to you later. Introduction to the digestive system and nutrition. Digestion refers to the mechanical and chemical breakdown of foods and absorption of nutrients. The digestive system carries out the process of digestion. There's two kinds of digestion here that I want you guys to know and be familiar with. First one there is mechanical digestion. Involves the breaking down of large pieces of food into smaller ones. Chemical composition is not changed by this process. The whole idea of chewing your food, called mastication, okay, M-A-S-T-I-C-A-T-I-O-N. This is the process there of chewing your food. And if a chewed banana has the exact same chemical composition as a banana that's just been peeled, okay? To alter the chemistry of it, now we're into chemical digestion. Breaking down of large nutrient molecules into smaller ones by breaking chemical bonds. And it actually begins in the mouth with we produce an enzyme. Our salivary glands produce amylase. And amylase is an enzyme that breaks down long carbohydrate molecules like starch into their smaller constituents, glucose, these kinds of things. But we'll get there. So mechanical digestion, chewing, chemical digestion, enzymes in the mouth, the stomach, breaking down chemically. The digestive system consists of the alimentary canal. And basically, this is from rooter to tutor, from mouth until anus, okay? And there are several accessory organs in between that assist in the process of digestion. So here's your alimentary canal, okay? And basically, it is just, it's a one big lumen, L-U-M-E-N. A lumen is just a tube. And it can be removed completely, the whole digestive tract in one shot, okay? And you have a look here. There's lots of good stuff here. We're going to get into the salivary glands, the mouth, the pharynx, the esophagus, the stomach, small intestine, large intestine, rectum, anus, pancreas, gallbladder, and liver. We're going to talk about all these, so sit back and enjoy. The alimentary canal, muscular tube that passes through the thoracic and abdominal pelvic cavities. It is the passageway for food between the mouth and the anus. It's about 8 meters long, so about 32 feet. Okay? The alimentary canal is about 8 meters long. The tongue to duodenum is about a half meter. The small intestine is about 6 meters in length, and the large intestine is about 1.5 meters in length. And something common to the entire alimentary canal is it basically has the same 4 layers throughout, all the way from rooter to tutor. The inside layer, or if you want to start with the outside, there's your serosa layer, muscularis. I wonder what that is for. Maybe a little peristalsis, muscle. And then you have the submucosa and the mucosa. So if you ever get caught in a pinch and you need to memorize these, MSMS, okay? If you start with mucosa, submucosa, muscularis, and serosa, however you want to learn it, but it needs to be learned. And layers of the wall of the small intestine. We're kind of jumping ahead here a little bit, but just to show you there that those same 4 layers are there. Okay, throughout the entire alimentary canal. And you'll see here that if you go into the inside here, there's your mucosa layer. Okay, and then you have the submucosa layer. Okay, on the there, and then, sorry, just trying to get out of here. There is your serosa layer, and here on the in between the submucosa and the serosa, there's your muscularis layer, circular muscle, longitudinal muscle, and you see they're going different ways, just for the whole idea for peristalsis. We're going to be introducing you there to this concept of of a lacteal. And you can see what's going on here. You've got your cardiovascular system as well as your lymph system. As we know, wherever there's cardiovascular, you'll find the lymph. And this is on the inside of the small intestine. This is how the small intestine absorbs. Okay? And you'll see there that we have there be villi. These are villi, and then you on top, if you can actually go on, you have what's called microvilli. Okay, so it's all a matter of surface area. And you think there, how can your body digest and absorb a Big Mac combo, in such a small area of the small intestine? Because the majority of the absorption happens in the duodenum there, the first part of the small intestine, which we'll get there. But you figure there, there's not a lot of space. How can we absorb everything there that we need? Well, if you flatten out the entire surface area there of the absorption area, you're looking at about the size of a tennis court. Huge amount of surface area. And we'll get into that. The body is a marvel of technology for sure. Great little introductory slide. Shows you all the 4 different layers there that are involved. There's your serosa, muscularis, submucosa, mucosa. And those 4 layers will carry throughout the entire course of the presentation. The structure of the wall of the alimentary canal, as we said there's your mucosa, submucosa, muscularis, and serosa layers. The mucosa layer, the inner layer of the wall, a mucous membrane that surrounds the lumen of the tube, consists of epithelium, underlying connective tissue, and a little smooth muscle, protects the tissue of the canal, carries on secretion and absorption of dietary nutrients. In some regions, the mucosa is modified into folds or projections to increase the surface area. Submucosa lies under the mucosa layer, consists of loose connective tissue, houses blood and lymphatic vessels, nerves, and glands, nourishes the surrounding layers of the canal, and vessel transport absorbs nutrients away from the digestive organs for sure. Muscularis, consists of 2 layers, as the name implies, of smooth muscle, inner circular fibers, and outer longitudinal fibers. Propels food through the canal through peristalsis. Serosa, the outer layer or visceral peritoneum, protects underlying tissue and secretes serous fluid to reduce friction between the abdominal organs. All right, so you'll see those 4 layers there throughout, so don't be surprised. Moving on, we'll start off with the mouth. Ingest food, mechanically breaks up solid particles, mixing them together with saliva. This process there is called mastication. Functions as the organ of speech and sensory reception. Surrounded by lips, cheek, tongue, and palate. Includes the oral cavity and vestibule. All right. And we're not, this is part of the digestion. We got to do it. But, I'm not going to go too heavy on, dental, which we do cover. We're going to look at a tooth in a little bit of complexity. The tongue is a thick muscular organ that occupies the floor of the mouth and nearly fills the oral cavity when the mouth is closed. Parts there that you might want to know: lingual frenum, papillae, and lingual tonsils. Okay, there's the root of the mouth there. There's the body. There's the papillae, palatine tonsils, lingual tonsils there, and epiglottis there at the back. The teeth are the hardest structures in the body. They are primarily deciduous teeth, numbering around 20. There are secondary permanent teeth numbering 32, and you're looking at dentine, right? A tooth consists of a crown projecting beyond the gum and root, which is connected to the jawbone. The crown of a tooth is covered by enamel, which consists of calcium and is the hardest substance in the body. Under the enamel is dentine, a substance even harder than bone. The central pulp cavity of a tooth contains blood vessels and nerves. Absolutely, teeth are alive. A layer of cementum connects the tooth to its periodontal ligament, which then connects it to the jaw bone. And all of that right here, you'll see that this is the root, this is underneath the gum, this is the crown, this is the tooth there that we see, it's above the gum. There's your enamel and your dentine, okay, pulp within the pulp cavity, and you'll see that all the blood vessels in there very much alive, okay. Gingiva, that's the edge of the gum there that touches the tooth. And if you're not familiar, you will be now. There is a disease there associated there when if the gums start to recede. It's called gingivitis. Okay, so it's very important to brush and clean your teeth, but it's also very important to brush and clean your gums as well. Okay, there's your root canal, periodontal ligament in here that holds the tooth to the bone, and then there's your cementum right in there. Okay. Just a little recap there. Cheeks, lips, tongue, palate, and teeth. Location and functions. That is definitely a good one. Have a read of that one. Pause it and have a look. The salivary glands, okay, and they produce saliva, which contains enzymes that begin chemical digestion of carbohydrates, and this is amylase, okay? As well as bicarbonate ions to keep pH favorable for enzyme activity and to protect teeth from acidic foods. There are 3 pairs of salivary glands. Okay, you've got the parotid, submandibular, and the sublingual under the tongue there. Okay. The different salivary glands have varying proportions of 2 types of secretory cells. We've got serous and mucus. Serous cells produce a watery fluid with a digestive enzyme called salivary amylase, which splits starch molecules into disaccharides. Okay, breaks down bread into its sugars. And a little trick there, if you have kids, it's you can do a little experiment there with them, or you can do with yourself there. Take a piece of bread and you chew a couple of pieces of it, or a couple of bites of it, or don't stuff the whole thing in your mouth, you might choke, but take a couple bites of just plain old-fashioned white bread and chew it. Don't swallow it, chew it for like 5 minutes, and you'll see there that this bread actually starts to turn sweet. It turns into this disgusting gelatinous mixture there that's kind of hard to hold in your mouth, but if you just keep it in there for about 5 minutes, you'll see that this amylase starts to break down the starch in bread into sugar, and that's all bread is. It's huge calories because it's just white table sugar. Okay, so there is your serous cells. They produce amylase as well as mucus cells that they surprisingly secrete mucus. Moving on down here, we're into the pharynx and the esophagus. Okay, we're down into this area right in here. And sorry, I'll see if I can kind of blow this up a little bit. And I move this over here. There we go. Move that up. And there you go. Good. So the pharynx is a cavity lying posterior to the mouth. Okay, so this whole area here is a better shot there coming up there the next slide, but just wanted to show you that it's the whole area, okay, the pharynx. And the esophagus is a muscular tube leading down. And, we've already done respiration. And respiration, there's your trachea, right? Epiglottis folds down, the food goes down the esophagus, but if you're not eating any food, the air can just go and hit the trachea, okay? The structures of the pharynx there is broken up into 3. You've got the nasopharynx, the oropharynx, and the laryngopharynx. Okay, naso is up top, oryngo, okay, it's for food right in here, this area, and the laryngo is down in here. I'll show you guys a better slide there, next slide. And you'll see that nasopharynx, it's just, this whole area is called the pharynx, but they've broken it up into 3. Naso, because it's related there to the nose, the nasal area. Oropharynx in here, and then we have the laryngopharynx in here. Okay, just another shot. I just put this in here. It's always cool to see a cadaver, isn't it? There you go. There's your 3. Perfect. The movement of the tubes, and you're gonna, I put this in here because peristalsis, you encounter it there when you drink a glass of water. So your esophagus is involved in peristalsis. Your stomach, when it rumbles or moving food around, that's peristalsis. When you're talking the intestines, they're moving things around, and that's peristalsis. So I had to put that in here at this point in time. The motor functions of the alimentary canals are of 2 types. Okay, mixing movements. Okay, there's segmentation and then propelling movements, right? So the mixing movements there occur when the smooth muscle contracts rhythmically in small sections of the tube. You see what's going on here? There's your stomach, there's your intestine. So there's what we call segmentation. In the small intestine there, mixes by alternately contracting and relaxing the smooth muscle in each area. So you see, let's say here's a food parcel, and it separates it. It moves it all around. It squeezes it. And it's all about surface area, right? And coming in contact with that food particles so that it can digest and absorb it. Okay. As well, there's the propelling movement. Okay. Including a wave-like motion. Okay. And peristalsis, a ring of contraction in a portion of the tube occurs while relaxing occurs ahead of the ring. This allows a mass of food to be propelled to the next segment. Here we're talking about the esophagus moving food around. So mixing movement, segmentation, and peristalsis. Okay. So the esophagus is a muscular food passageway from the pharynx to the stomach, about 25 centimeters. Goes through the diaphragm, okay, through the esophageal hiatus, mucus glands in the submucosa. Lower esophageal sphincter surrounds the lower end, also called the cardiac sphincter. Okay, there's your layers. There's just a shot of what the esophagus looks like there from a cross section. And in a lot of ways there, it just kind of looks like a breakfast sausage. Some of you guys, if you've ever dissected a heart and then there might be a piece of esophagus on there, I know we do that in the lab there, and it looks just kind of like a breakfast sausage there. Not like the trachea, which had those cartilaginous C-rings there, very protected. The swallowing mechanisms there, there's 3 stages there. There's the voluntary stage, okay, sensory reception stage, and then the peristalsis stage there. And basically, we only have control over this one. Once you swallow a banana, you have no idea what's going on. You don't see it again for about 36 hours. Okay, so first stage there, voluntary stage, where the food is chewed and mixed with saliva, forming a bolus. The bolus is forced into the oropharynx with the tongue. Okay, and you'll know there that the whole act of swallowing, the epiglottis comes down, protects the trachea, and that food bolus goes down into the esophagus, right? So sensory receptors of the pharynx sense food, okay, triggers the swallowing reflex, okay? And I'm just going to put an E in there anyway. There you go. The soft palate and uvula elevate to block the nasal cavities because we don't want food going up into the nose. The hyoid bone and the larynx elevate, and the epiglottis closes off the larynx, so no food goes down the trachea. Tongue presses against the soft palate to seal the oral cavity from the nasopharynx. The pharyngeal muscles contract, pulling the pharynx upwards. Muscles of the laryngopharynx relax, opening the esophagus. Peristaltic waves begin in the pharynx and proceed towards the esophagus. Breathing is inhibited for a brief moment. Stage 3, peristalsis transports the food in the esophagus to the stomach. There you go. Pretty easy peasy. Moving on there to the stomach, it's kind of a J-shaped muscular organ in the upper left abdominal quadrant. Okay, you'll see these ridges within. These are called rugae or gastric folds. And again, it's all about surface area. You're increasing the surface area of the stomach by these three-dimensional folds. Also, they give traction there so that they can throw food around, that kind of stuff. Receives food there from the esophagus, mixes food with digestive juices, begins digestion of proteins. Okay, the digestion of carbohydrates already began up in the mouth there with the amylase. Now with the hydrochloric acid and the other stuff there that we'll get into, you'll see that it begins the digestion of proteins, which is they're pretty harsh. Proteins are hard to digest because of the nitrogen. Okay, limited absorption of nutrients occurs in the stomach, propels food to the small intestine, and you just be careful there when you're talking an ulcer. Okay, H. pylori is a bacteria there that they used to think that ulcers were caused by spicy food or smoking or excess alcohol intake, but as it turns out, it's a bacterium. Okay, H. pylori bacterium there causes an ulcer, and we'll get into that. But it actually it causes a hole in the stomach, so acid food contents can actually leak out into the outside of the stomach. It's quite severe, actually, painful. The stomach is divided into the following regions. Okay, you have the cardia region, okay, a small region near the opening of the esophagus. The fundus region. Do you know any other organs there that have a fundus? Well, you'll have to wait till week 13 when we do reproduction. You'll see there that the uterus, okay, also has a fundus, this area jetting up, okay. The body region in here, main portion of the stomach between the fundus and the pylorus, which is down here. This is called your pylorus region, okay. The pyloric canal is a narrowed area close to the small intestine. The pyloric sphincter, okay, is a ring of muscle there at the bottom of the stomach that opens and closes as food at this point in time is called chyme, C-H-Y-M-E. This is food there that's been mixed with the hydrochloric acid and it's ready to come out to be digested by the small intestine. Okay, so there's your lower esophageal sphincter, okay, right in here. I've seen this also called the cardiac sphincter, and this is your pyloric sphincter. You'll see there that the lining of the stomach, there is lots going on. Okay, you've got these gastric pits, and you see here's a real electron micrograph, and here's the cartoon. I kind of like seeing both there. There's your same mucosa, submucosa, muscularis, and serosa. Okay, but you're going to see these gastric pits, and that's where all of these, it's all about surface area, right? And you'll see what's going on here. These gastric pits, there's your mucus cells, parietal cells, chief cells. Okay, there's your mucus cells there. There's your parietal cells, these guys in here. Okay, and here's your chief cells. We're going to hit those right now. Okay. The gastric glands within the thick mucosa of the stomach open into the lumen as gastric pits. These gastric glands generally contain three types of secretory cells. There's mucus cells, goblet cells, produce mucus that protects the stomach lining, because you have to think the stomach is coated in hydrochloric acid, pH of about 1 or 2. This will eat through a table. Now, the only thing that's protecting your stomach and your body from this hydrochloric acid is this mucus. All right. Chief cells secrete inactive pepsinogen, which is activated when it comes in contact with hydrochloric acid and changes to pepsin. Now it's ready to digest protein. Parietal cells, these guys here, they secrete hydrochloric acid, which activates the pepsinogen, okay, as well as intrinsic factor, which is required for B12 absorption from the small intestine. Okay, so mucus cells, goblet cells, chief cells, parietal cells. Okay, very good. And just a little recap here of what's going on. There's your pepsinogen, chief cells, inactive form of pepsin. Pepsin here, okay, pepsinogen plus hydrochloric acid, and it's an enzyme there that's able to digest protein. hydrochloric acid produced there by the parietal cells, mucus cells there, the goblet cells, and intrinsic factor source there, parietal cells of the gastric glands necessary for B12 absorption. Okay. There are 3 phases of gastric secretion, and like when you walk by a Kentucky Fried Chicken and you smell that glorious grease. Ugh, does smell good though. But and it gets your mouth watering, gets you thinking about food. That's the whole point. We actually call this the cephalic phase of digestion. The sight, taste, smell, or thought of food triggers parasympathetic reflexes. Gastric juices get flowing, right? That's the whole point of an hors d'oeuvre, right? It's a little bit of meat on cracker or cheese on cracker, just a little bit, just enough just to get things going, and then you turn into a raging, eating beast, right? Next phase there, gastric phase. Food in the stomach chemically and mechanically stimulates the release of gastrin, which in turn stimulates secretion of gastric juices. Reflex response also stimulate gastric juice secretion, so now the stomach is involved. Intestinal phase, as the food enters the small intestine, it stimulates the intestinal cells to release intestinal gastrin, which in turn briefly promotes the secretion of gastric juices. Now we're talking intestinal digestion. Okay, we'll get there, but these are the 3 phases, right? Cephalic, gastric, and intestinal phase of digestion. You will remember the sympathetic and parasympathetic. Okay, sympathetic is fight or flight. Okay, if your life is in danger, you don't need food. All right, so all of the digestion is not involved with this system at all. The part of the nervous system that is directly related to digestion is parasympathetic. Okay, rest and digest. Impulses increase gastric activity. Homeostasis and hormones which affect gastric secretion include somatostatin, inhibits acid secretion. Okay, gastrin, which increases gastric juice secretion, and we've got CCK, cholecystokinin, CCK. All right, cholecystokinin released by the small intestine cells when protein and fat enter the small intestine. Decreases gastric motility. We'll get more into these as we go on. Okay, here's a little bit of homeostasis, regulation there of gastric secretions. You'll see here that an impulse conducted by the parasympathetic preganglionic nerve fiber, okay, vagus nerve, okay, and then parasympathetic postganglionic impulse stimulates the release of gastric juices from the gastric gland, okay. Now, number 3, impulse stimulates the release of gastrin into the bloodstream, which in turn, gastrin stimulates the glands to release more gastric juice. So this is just the whole idea, and if you want to relate it to here, cephalic, gastric, and intestinal, this is the whole thing of the regulation and secretion there of these gastric juices. Okay? Mixing and emptying, okay? The mixing of food in the stomach with gastric juices produces a semi-fluid paste called chyme. So your food comes in, you just chewed it, and then it mixes all in here, and you'll see, and now it's all broken down into this food slurry, completely dissolved, and then it's now released into the duodenum for absorption. Pyloric sphincter relaxes, and the food squirts in. Chyme. The enterogastric reflex slows the stomach emptying. And just to zoom in here, you'll see there that the duodenum fills with chyme. Okay, we just ate and it mixed around, and now that chyme is coming into the small intestine. The sensory stretch receptors are stimulated. You see, it stretches, right? And that tells us there that there's food that's entering, okay? And the sensory impulses there, they conduct there to the central nervous system, okay? To the central nervous system. Hey, that's right, something's going on here. So a reduction in the parasympathetic activity decreases peristalsis in the stomach wall. And just the whole point is you've got food already in here, so just take it easy. That's enough. let that sit in here, let this food go down, and then once this food goes down, the stretch receptors will decrease, okay? They will not be stimulated, so therefore now we're ready to go for another shot, and this whole process will continue, okay? So a little bit at a time, shut it down. A little bit at a time, shut it down until all of this is moved on, okay? So that's mixing and emptying actions there. Now that we're, you can kind of see we're kind of progressing, okay? We brought food in, we started off at the teeth and amylase, and we went down to the three pharynx and peristalsis down the esophagus, through the sphincters here, and through mixing and opening of the pyloric sphincter, and the chyme is now down in here, right? Like, this is very acidic, okay? And the stomach can handle the acidity because of these mucus cells. But the intestines aren't producing that mucus as much, okay? So they don't have the protection. So the small intestines cannot handle the acidity coming out. So when you get into this one here, this whole idea, this is a very important process because this signals that there's highly acidic food right in this area, and the small intestine can't handle it. So this is where we get into there the pancreas, and you'll see the location of the pancreas in conjunction with everything else. There's your duodenum. You'll see other videos there. They'll call it duodenum, however you say it. The beginning of the small intestine right there. And the pancreas has a dual function. Okay, it's both endocrine and exocrine. And endocrine, there is no duct where a hormone is made, and then the hormone just kind of reverberates through the interstitial tissue until it's eventually picked up by the bloodstream and moved. Okay, an exocrine, okay, gland. This one here has a duct. Okay, and it has something is produced, and then it is jettisoned through this duct to where it needs to go, squirted, that kind of thing. So the pancreas dual function, endocrine and exocrine. The exocrine function of the pancreas is to produce pancreatic juice that aids in digestion. Okay, and that's where we're at going right here. Okay, and you can see there that we have a shot of the gallbladder as well in this particular slide. And you can see here the bile duct comes in, and it actually meets up, okay, with the pancreatic duct here right in here. And you'll see that there's an opening, if I can zoom in without destroying my PowerPoint here. You can see what's going on here. So whatever is being made here by the pancreas can be squirted out and jettisoned out right into the duodenum as this food comes in, this highly acidic food. And you'll see there that the pancreas produces many things. One of them there is basically baking soda, okay, bicarbonate ion that is able to increase the pH, because this chyme coming in has a very, very low pH, probably about 2, which will damage the intestine and wall for sure. So that pH needs to be brought up, and that's what it does. It squirts baking soda right in here and raises the pH. Structure of the pancreas, closely associated there with the small intestine, as we said. Pancreatic acinar cells make up most of the pancreas. These cells produce pancreatic juice. Okay, acinar cells cluster around tiny tubes that merge to form larger ones and then give rise to the pancreatic duct. Okay, the pancreatic and bile duct join and empty into the duodenum of the small intestine together. The hepatopancreatic sphincter regulates passage of the pancreatic juice and bile into the duodenum. Okay, and that's what we call the hepatopancreatic sphincter. Right in there allows the bile and the pancreatic juice to come on out. And there's a close-up shot of it right there. Okay, bile duct, pancreatic duct, hepatopancreatic sphincter right in here. Perfect. Pancreatic juice is not just one thing. Contains enzymes that digest carbohydrates, fats, proteins, and nucleic acids. Pancreatic amylase breaks down complex carbohydrates, starch, and glycogen if they haven't already been broken down already. Pancreatic lipase, fat breakdown, right? Breaks down triglycerides to glycerol and fatty acids. Two nucleases break down the nucleic acid to nucleotides. Proteolytic enzymes, trypsin, chymotrypsin, and carboxypeptidase each split certain bonds between amino acids to break down proteins to dipeptides. So these right here, trypsin, chymotrypsin, these are the ones there I'd like you to know as far as protein protein breakdown. Proteolytic enzymes are secreted in active forms and stored in zymogen granules. Upon reaching the small intestine, they are activated. For example, enterokinase converts trypsinogen into active trypsin. This prevents these enzymes from digesting the pancreas. We don't want these enzymes to start breaking down the pancreas. We send them out, and in the presence that they'll turn into what we need them to turn into, where we need them in the small intestine. Pancreatic juice also contains bicarbonate ions, which creates an alkaline environment in the duodenum there for enzyme function, okay, to counter the acidic conditions there from the stomach. Regulation of pancreatic secretion by secretin. Hormones which help regulate the release of pancreatic juice include there's secretin, stimulates pancreas to release pancreatic juice high in bicarbonate ions, and we also have cholecystokinin, secreted there by the small intestines, stimulates the pancreas to release pancreatic juice high in enzymes. And you'll see what's going on here. We'll just blow this up just a little bit there. So you have the acidic chyme enters the duodenum, okay, right in here. The intestinal mucosa releases secretin into the bloodstream. Okay, those sensors there, stretch receptors, realize there's something there. Now, the intestinal mucosa releases secretin into the bloodstream. The secretin stimulates the pancreas to secrete bicarbonate ions. Okay, pancreatic juice rich in bicarbonate ions passes down the pancreatic duct to the duodenum, down here, where the bicarbonate ions neutralize the acidic chyme. Okay, that's how the whole thing goes in a nutshell. There's a beauty movie for you to watch as well. The pancreas has two major kinds of secretory cells. That is why it's a dual functioning organ. They're exocrine and endocrine. There are three important hormones there that are secreted by the islets of Langerhans. And these are these specialized cells there that produce these hormones. Great movie. Now we'll get into a little bit of diabetes, these kinds of things here. But you'll see that alpha cells, they produce glucagon. This one here is involved with sugar. Beta cells are involved with insulin. Again, dealing with sugar. Okay, they are opposing. Okay, insulin forces the cells to absorb sugar. Glucagon kind of pulls the sugar off the shelves and dumps it back into the blood. There are also delta cells that produce somatostatin, and this is for carbohydrate regulation. Okay. The pancreatic hormones work as a team to maintain homeostasis of food molecules. There's glucagon, tends to increase blood glucose levels and to stimulate gluconeogenesis in liver cells produced by the alpha cells. Okay, it's kind of the opposite of storing sugar. We take the sugar that's been stored on the wall, we modify it, and we throw it back into the blood. They're raising the blood glucose level. Insulin lowers the blood concentration of glucose, amino acids, fatty acids, promotes their metabolism by tissue cells, and these are produced by beta cells. Okay. We'll get more into this one there when we get into type 1, type 2 diabetes for sure. Ghrelin GHRL stimulates the hypothalamus to boost the appetite, slows metabolism, and fat burning, may contribute to obesity. Somatostatin primary role is regulating the other endocrine cells of the pancreatic islets produced by delta. This one here takes care of all the other cells. So this is a very important one for sure. Pancreatic polypeptide influences the digestion and distribution of food molecules to some degree. Okay, not so worried about this one here, but that one for sure, and these two 100%. Yes, yes, yes. Did I just say that? Good movie there on the pancreas. The whole idea of being able to raise and lower your own blood sugar. There's a lot of people there that can't. You've got the type 1 diabetes, which is genetic, okay, generally diagnosed in younger individuals, okay. They can't produce insulin, so they need the shot every day. Type 2 diabetes, generally diagnosed in older individuals who didn't eat as well, or their lifestyle, they still produce insulin, but their cells don't recognize it anymore. So they type 2 diabetes, they are what we call insulin resistant. Good little slide here, alpha cells, beta cells. Have a look at that. Let me know if you have any questions. Type 1 diet, actually before we get there, blood glucose levels rise, pancreas secretes insulin. Blood glucose levels fall, pancreas secretes glucagon, okay, to pull that sugar off the shelf and dump it back in the blood, raising the blood glucose level there. Type 1 diabetes there is an autoimmune disease where the immune cells attack the pancreatic beta islet cells that produce insulin. So this person here cannot produce any insulin, okay? Usually diagnosed in children 8 to 10, somewhere in that range. Type 2 diabetes, these individuals, they produce insulin, but due to poor lifestyle change, lifestyle routine over the long period of time, their body becomes resistant, okay, to insulin. So what happens there, the person there eats a piece of cake, and the insulin there doesn't work for the cells, so the sugar stays in the bloodstream and doesn't enter the cells. So essentially these people starve. Okay, symptoms: increased hunger, high blood sugar, weakness, fatigue, dizziness, fainting for sure. Okay, the general functions here, now we're talking about the liver. This one here, the liver, this one is a super organ. Just some specific functions: carbohydrate metabolism, okay, lipid metabolism, protein metabolism, storage, blood filtering, detoxification, secretion. And we're not even done yet. Okay, metabolic activity produces glycogen from glucose, breaks down glycogen into glucose, converts non-carbohydrates to glucose, oxidizes fatty acids, synthesizes lipoproteins, phospholipids, and cholesterol, converts carbohydrates and proteins into fats, deaminating amino acids, forms urea, synthesizes plasma proteins, converts some amino acids to other amino acids, stores glycogen, iron, and vitamin A, D, and B. Yeah, you got to be careful that it will store these fat-soluble vitamins, but you have to be careful. There was a loving mother who maybe was working late, and the kid would come home there from school and would eat those gummy vitamins like candies. And the problem with those gummy vitamins isn't the water-soluble vitamins. The child just peed them out. It's the fat-soluble ones that they end up being absorbed by the liver, and they stay in the liver. And now this 11-year-old kid has the liver of a 90-year-old drinker. So just be careful, okay, of phagocytosis of worn out RBCs and foreign particles, removes toxins such as alcohol and certain drugs from the blood, acts as a blood reservoir. So basically what the liver does is, we're up here, and the whole idea, the food comes down the stomach and into the small intestine where it's absorbed, okay, by those lacteals. And now all the food there that has been absorbed by the small intestine, where does it go? Directly into the bloodstream? No. All of that food that just absorbed by the small intestine, what happens now is all of that food goes directly to the liver. where is detoxified, cleaned, inspected, and then it goes through to the bloodstream. Okay, so the liver is the intermediary between all your food and the blood. Anything there that gets into your blood has got to go through the liver first. It's got to be detoxified. Okay. So, yeah, the liver's pretty important. Structure of the liver there, blood from the hepatic portal vein carries blood rich in nutrients to the liver, this coming from the small intestine. A portal system, okay, has capillaries at two ends. So you'll see there that the capillaries of the lacteal of the small intestine, they absorb the food, and then that food nutrients travels through the hepatic portal vein directly to the liver, where again there are capillaries there ready to exchange and get that food back into, or get that food into the blood. So a portal system, you have capillaries on both ends, and the hepatic portal system, this carries food from the small intestine to the liver for detoxification to clean it all up before it goes into the blood. Blood passes from the sinusoids into the central vein of a lobule and exits the liver via the hepatic vein. Cooper cells carry on phagocytosis in the liver. Bile and secretions from hepatic cells are collected in bile caniculi, which empties into the bile ductules that converge to become a hepatic duct and finally form the common hepatic duct. The common hepatic duct transports bile out of the liver. And there's a nice movie. Pause the video, watch that one there. So your liver lobules here, okay, the whole idea that this is coming from the small intestine. This is your hepatic portal vein, and that food coming from the small intestine comes up here, goes to the liver, and you'll see there that there's the whole idea of capillaries, right? Capillaries are the only place where things can be exchanged. So all that food came from the small intestine. Now it's going through the hepatic portal vein, comes into here. You see these lobules here. These are the ones there that the food, capillaries here, the food has got to travel through, get cleaned up, and then it goes on to the bloodstream. Okay, that's how it works. These liver lobules and the bile formed from blood in the liver lobules there assist in digestion of fats stored in the gallbladder. Here we're talking bile, composition, water, cholesterol, and bile pigments there. The hepatic cells use constituents of the blood to manufacture bile. That green structure, okay, in the illustration represents the bile carrying duct right in here, okay, and that's what's going on. These tiny ducts are called bile caniculi. They drain into the larger ducts, okay. Just another shot here, the flow in the liver lobules, okay. There's your hepatic vein, and you'll see what's going on in here, right? Your bile is coming down. Here's your portal triad, and it's a beautiful slide. Have a look at it. Let me know if there's any issues. The more on the hepatic lobules, you'll see there that the bile formation, okay, bile is made in the liver, and it comes down here, and you'll see there that this is blood coming from the small intestine for filtering, okay. Kind of moving in the opposite directions here. All right. Another shot of the same thing. There's your bile duct, and there's your branch of the hepatic artery. Food is coming up here for the whole point of being filtered and cleaned. And there's your central vein, blood flow out of the liver. Okay, so it's moving up this way here. Okay, good. Composition of bile. Bile is a yellowish green liquid that hepatic cells continuously secrete. Bile contains water, bile salts to emulsify fats, help absorb fatty acids, cholesterols, and fat-soluble vitamins. Bile pigments, bilirubin and biliverdin. You might remember those ones there from blood. Cholesterol, electrolytes, and only the bile salts have a digestive function there. Okay. The gallbladder is a pear-shaped sac on the inferior surface of the liver. This is where the bile storage occurs, and this happens there between meals and reabsorbs water to concentrate the bile. Contraction of its muscular wall releases bile into the duodenum. You remember there when we saw the, sorry, what slide is that? 50, just so I don't lose it. And you remember there, there's your bile duct. And that's how the bile gets right in, right beside the pancreas. It's connected there to the cystic duct, which joins the common hepatic duct to form the common bile duct. The common bile duct leads to the duodenum, where it joins the pancreatic duct. The hepatopancreatic sphincter controls the release of bile from the common duct. In certain conditions, cholesterol may precipitate in the gallbladder to form gallstones, which have to be removed. The gallbladder is pear-shaped. Yeah, and what I wanted to show you here, these are some beautiful gallstones right in here. Wonderful, wonderful, wonderful. Okay. Regulation of bile release. Now, you'll see here, I'll zoom in a little bit. Bile does not normally enter the duodenum until the hormone cholecystokinin, CCK, stimulates the gallbladder to contract. Proteins and fats in the small intestine stimulate the release of CCK. The hepatopancreatic sphincter remains contracted until a peristaltic wave approaches it, at which time it relaxes and a squirt of bile enters the duodenum. And you can see what's going on here. Oh, sorry. The chyme with fat enters the duodenum. Cells of the intestinal mucosa secrete the hormone CCK into the bloodstream. Okay. And that goes into the bloodstream and eventually makes its way. CCK stimulates the muscular layer of the gallbladder wall to contract. Squeeze, squeeze. Bile passes down the cystic duct. Okay. And the hepatopancreatic sphincter relaxes and bile enters the duodenum. So by the time fat hits here, the bile has already been released and it's ready to go. Okay. Moving on into the small intestine there, a long tubular organ which runs from the pyloric sphincter of the stomach to the beginning of the large intestine, fills a large part of the abdominal cavity. Functions of the small intestine include release of chyme from the stomach, receives chyme from the stomach, receives pancreatic juice from the pancreas and bile from the liver and the gallbladder, finishes digestion of nutrients that arrive in the chyme, absorbs digestion end products, transports the remaining residue to the large intestine. Okay, lots going on here. The small intestine consists of the duodenum, jejunum, and ileum. The duodenum is the shortest and most fixed portion of the small intestine. The stomach empties chyme directly into it. The rest is mobile and lies free in the peritoneal cavity. The jejunum makes up the approximately 2/5, about 40% of the small intestine, and has a slightly larger diameter than the ileum and is more active in the digestion process. The most distal portion of the small intestine is called the ileum. That's the last. The jejunum and ileum are suspended from the posterior abdominal wall by the double-layered fold of peritoneum called mesentery. Okay, kind of like a fat blanket. The mesentery contains blood vessels, nerves, and lymphatic vessels that serve the intestinal wall. The greater omentum covers the intestines. This is your fat blanket for sure, a double fold of peritoneal membrane. Okay. All these other shots there were kind of small, but just have a look here of what's going on. There, your stomach, okay, pyloric sphincter. There's your duodenum, okay, and eventually there we make it to the jejunum, and then your ileum there is at the end, and it connects the small intestine there to the large intestine. This is just a shot. I just wanted to show you there that fat blanket, the greater omentum, just holds everything there together, keeps your small intestines in place. The mesentery suspends the small intestine, and you'll see there that all of these little organs, there's your, you see, here's your greater omentum, okay? Covers everything here, but everything has its place. Everything is packaged up. Everything has its place. You do need fat because it holds everything together, okay? Structure of the small intestinal wall. The inner wall of the small intestine is lined with finger-like intestinal villi, which increases surface area, available for absorption. And like we talked about it there before, the body is a marvel at creating space where there is none. Each villus consists of a simple columnar epithelium with a core of connective tissue housing blood capillaries and lymphatic capillaries, and we call this a lacteal. We've seen those there before. We'll get back into them right now. Blood capillaries and lacteals transport absorbed dietary nutrients out of the alimentary canal. Between the base of the adjacent villi are tubular intestinal glands. Epithelial cells lining the small intestines are replaced about every 3 to 6 days, and we've seen this there before. This is what the inside there of the small intestine looks like, and it has these villi. Cardiovascular system, as well as you got it. There is the lacteal in all of its beauty, cardiovascular and your lymph, all right? And you'll see there that, how does the small intestine absorb everything? If you zoom in, you'll see on here what's called microvilli. So basically, you're just absolutely increasing the surface area by a huge factor. And we'll show you this there. So there are these microvilli that are on top. And, they're just basically mini lacteals. And it's all about absorption. Look at all the extra surface area there that we have. And there's your microvilli. Beautiful stuff. Just more shots of it again. There's your villi on top of the inside wall of the small intestine. And then you have your microvilli on top of those. And again, you still have your same layers. Mucosa, submucosa, muscularis, and serosa. Secretions of the small intestine. Mucus by the goblet cells, which are abundant throughout the mucosa. The mucus-secreting glands, they're located in the submucosa of the duodenum. Intestinal glands at the base of the villi secrete large amounts of watery fluid that carry digestive products into the villi. Epithelial cells of the mucosa have digestive enzymes embedded. There's pepsidases, which break down dipeptides into amino acids. You've got sucrase, maltase, lactase. You guys know there that anything that ends in ase is an enzyme. Didn't want to do that. All right, so breaks down disaccharides to monosaccharides there. Intestinal lipase, this is a fat. Breaking down fats, breaks down triglycerides to fatty acids and glycerol. Regulation of the small intestine secretions, mechanical and chemical stimulations from chyme cause goblet cells to secrete mucus and intestinal glands to secrete their watery products. Distension of the intestinal wall stimulates parasympathetic reflexes that further stimulate secretions there from the small intestine. Absorption of the small intestine. These villi increase the surface area. Absolutely, the small intestine absorption is so effective that very little absorbable material reaches the organ's distal end. So basically, by the time this food makes it to the large intestine, there's not much food left to be absorbed. Monosaccharides and amino acids absorbed through facilitated diffusion and active transport, absorbed into the blood. Fatty acids and glycerols absorbed by several steps involved as noted, absorbed into the lymph and blood. Electrolytes and water absorbed through diffusion, osmosis, and active transport right into the blood, all go into the hepatic portal system. There's a nice little recap. Absorption mechanism and means of transport. Good stuff. There's an example of a disaccharide. These are examples there of monosaccharides, the whole process of breaking down, right? Maltose through the enzyme maltase. Breaks these down into glucose and glucose. We've got a dipeptide here, 2 amino acids, and we snip it in half, dipeptidase, and now we have the individual amino acids. And through the use of a fat enzyme, lipase, we break this fat down into its individual fatty acids and glycerols for further breakdown. Okay. Movement of the small intestine. Again, we're back into peristalsis and segmentation. Okay. You want to go back up to the slide there earlier on. Go and have a look at that. Okay. Here's more of it here. There's your segmentation, peristalsis, moving things around and breaking them up. Okay. The large intestine is named large because its diameter is larger than the diameter of the small intestine, but it's only about 1.5 meters long. Okay. Begins in the lower right portion of the abdominal cavity. Okay. At the cecum, ascends on the right. Okay. Crosses over to the left and descends on the left-hand side, right? So starts over here. If we're looking at anatomical position, it goes up, across, and down. Opens to the outside of the body at the anus. The large intestine absorbs water and electrolyte and forms and stores feces. The cecum is a pouch at the beginning of the large intestine with the appendix projecting downward from it. Colon consists of the ascending, transverse, and descending. The rectum is a straight section of the large intestine which lies next to the sacrum. The anal canal opens to the outside at the anus, guarded by the involuntary internal and anal sphincter, and a voluntary external anal sphincter. And this is it right here. So you see there is the appendix. Here's your cecum. Okay, this is the end portion of the ileum, the small intestine. Okay, the food comes in through here, and then you'll see there's your ascending colon. Okay, transverse, and then descending colon, ending there in the anal canal and the anus itself. Okay, there's a shot there of the anal canal. Okay, and here is your external anal sphincter that we have control over. Internal, we do not. Structure of the large intestinal wall has the same 4 layers. Okay, mucus, submucosa. We've got the muscularis and serosa. And the walls of the large intestine do not have villi because they're not involved in food digestion. The large intestine is involved in water absorption and fecal compactment and housing there for our bacteria flora there that give us vitamin K in these ones here. Okay. Fibers of the longitudinal muscles are not uniform. Instead, they are arranged in 3 bands, okay, called tennae colae, that extend the whole length of the colon. These tennae colae exert tension, which creates a series of pouches or haustra in the large intestine. Okay, just a nice shot here, okay, of all your different layers, mucosa, submucosa, it's all here. Okay, and there's the lumen there of the large intestine. The large intestine has little or no digestive function, absorbs water, electrolytes, secretes mucus, houses intestinal flora, everything there that we've already talked about, fecal compactment, and carries out defecation. Okay? And the mixing and peristaltic movement of the large intestine are similar to those of the small intestine, but slower. We're at the end of the line here, right? These peristaltic waves, mass movement, occur only 2 to 3 times during the day after a meal in response to the gastrocolic reflex. Defecation is stimulated there by the defecation reflex that forces feces into the rectum where they can then be expelled. And feces are composed of material not digested or absorbed, water, electrolytes, mucus, bacteria. Bile pigments, okay, that have been altered there by bacteria provide the brown color. Okay, so mostly it's blood that's been altered there by the bacteria in the large intestine. The pungent odor is produced by bacterial compounds, including phenols, hydrogen sulfide, indole, skatole, ammonia, these kinds of things. Nutrition and nutrients is the study of nutrients and how the body uses them. These nutrients are chemicals supplied by the environment that an organism requires to survive. Required nutrients, carbohydrates, lipids, proteins, vitamins, minerals, water, yep. Macronutrients are required in large amounts and include lipids, carbohydrates, proteins. Micronutrients are required in small amounts, vitamins and minerals. Energy value of macronutrients is expressed in heat units called calories. Okay, fats provide about 9.5 calories per gram, while proteins provide about 4.1. The problem with proteins, like if you're ever in a fasting situation, the first thing the body will go for is carbs. Easiest for the body to break down. And if there's no carbs in your body, then your body will go after fats. Okay, you get a good return on your calories from fat. Good bang for your buck. The worst, though, is protein. In a fasting situation, your body will go after protein the least because it's very difficult for the body to break down nitrogen compounds. All right. Essential nutrients there are those that cannot be synthesized by human cells, some fatty acids, some amino acids, and some vitamins for sure. There's a beautiful slide on the summary of the major digestive enzymes there. Have a look at that. That is digestion in a nutshell. Please stop and start wherever you need. Other than that, enjoy your day, and we'll see you next week.